noticed by now, but we haven't talked about neurons very much. We've talked about the cells of the nervous system other than neurons, astrocytes, oligodendrogliomas, and when they become cancers, but neurons haven't talked about until now. So neurons, their function is to transmit signals to the nervous system. And the nervous system is based on signals. You get signals from the body to the brain, and you get signals from the brain to the body. Brain's telling the body to move your right arm. That's signals. It's all based on signals. So neurons have a very specialized structure to get this all done. They have dendrites all over the place, right here, here. These are to receive input from signals. Then they send the signals through the cell body into the axon, and the axon is the output area. So signals come in this way, and they come in this way, and they go out through the axon, into the axon terminal. The other thing to note is that cell bodies and the dendrites have Nissl staining. That's because they have rough endoplasmic reticulum, so they stain for that. Axons do not stain because they do not have any rough ER. Finally, neurons are permanent cells. They do not divide. That is why strokes can be so debilitating, because strokes is basically when your brain cells die, and when they don't die, they do not come back. So your neurological deficits can become permanent. However, you have injury to the axon, which is over here, maybe it gets cut here. If we do undergo something called Wallerian degeneration, then you can have regeneration of the axon. I'm going to go into detail about that now. You can see my very crude drawing of a neuron here. And we're going to talk about Wallerian degeneration. So I have a bunch of words here that are just copying what I'm going to explain right now. So ignore that for now. Look at the picture. So we have, let's say, transection of the neuron. And note, Wallerian degeneration only occurs in the peripheral nervous system. They try to happen in the CNS, but it doesn't work. So you can only get axon regeneration in the peripheral nervous system. So we transected it, right? It got cut, our axon got cut. What happens? Segment distal to the cut, far away from the nucleus, breaks down, okay? All of this breaks down, becomes debris, it's dead, okay? You cut off a limb, the part far away gets dead. The other thing that happens though is you also get degeneration this way, all the way to the closest node of Ranvier. So these are all the nodes of Ranvier. This is the closest one to the transection. So you're going to break down all the way up to this node of Ranvier. At the same time we have this degeneration, you're going to see something called the axonal reaction. You're going to get edema of the cell body. It's going to swell up. Okay, cell body swelling up. Your nucleus is going to move this way to the periphery. Okay, and you're going to see finally dispersed Nissl substance. From that's rough ER is making protein. This whole axonal reaction is basically showing you that you're making more protein synthesis that you're going to need for axonal repair, because your axons miss a ton of protein in it. So after all these things happen, your axon can slowly regenerate and you can make a new axon. Okay, and this takes time. Remember that. So you can actually regenerate your axon, but it only occurs in the peripheral nervous system, and this is Wallerian degeneration. And this axonal reaction, remember all the details here, is actually pretty key. There's the edema, there's the nucleus that moves out, and then there's a bunch of finely granular Nissl substance reflecting increased protein production. Alright, so we talked about how neurons send signals. And the question is, how do they do that? How do they send signals? And the answer is in the action potential. So the concept here is that we have a neuron. I'm just going to illustrate a neuron as a circle. And there's sodium channels and there's potassium channels, okay? Potassium channels go out, sodium channels go in, okay? At baseline, when the neurons is chilling, it's at a -70 because this one's very active, okay? This one's very active. It works a lot. You get K plus leaving, so it's more negative now, okay? So it's -70. And this one is not very active, okay? The sodium channel is not very active at this point. It's low permeability. So overall it's -70. So what happens is if you get a stimulus, it could be anything. It could be another nerve hitting this nerve. And it can cause depolarization. Basically increases this membrane potential above threshold. So this threshold here, -55, your membrane potential increases from -70 to above 55. You're going to cause a chain of events. You have these voltage gated sodium channels here. Okay, it's voltage gated, so they only open once you're above a certain voltage. Once you're above negative 55, these are going to open, and then when they open, you're going to get an influx of sodium. Lots of positive charge in here. Okay, and then so your voltage is going to go up. You're just putting a lot of positive charge. It goes up to about plus 40. You don't have to remember the numbers. Remember, it goes up because of the voltage gated sodium channels. Once you get to about a certain level, your voltage gated sodium channels are going to close. And then now you have some voltage gated K plus channels, okay? Efflux channels, these are voltage gated. Once you hit this certain level, they're going to open up, okay? Now your sodium can leave, your potassium can leave, and then once your potassium leaves, again, you're losing all this positive charge, so it's going to go back down, and you go back down. It's called repolarization, okay? Well, once you hit a threshold, this is going to be fourth stage. Your potassium channels are actually going to remain open for a little bit longer. So it's going to keep going down because your potassium's leaving, so you're losing all that positive charge. It's called hyperpolarization. You're basically overshooting your threshold, your resting membrane potential. Finally, they're eventually going to close the potassium channels, and then you're going to reach equilibrium state, which is right here at negative 70. Note that we have a whole axon here, okay? This is our cell body, this is dendrites, okay, and this is the axon. All this thing happened at one specific location, okay? This all happened here. This is our little action potential here. Alright, so what this is going to do though is once we get this peak up here, this is a positive, right? That's gonna influence this right here. It's gonna cause this side to go above threshold, and it's gonna shoot up and do its action potential. And then when this part hits its peak, it's gonna influence this side, and it's gonna cause this side to go above threshold because it's positive, right? So it's gonna make this side go above the peak, above the threshold, and it's gonna shoot up, and you're gonna go all the way down, okay? The thing is, this is kind of slow. So the way we can make it faster, because you got to do this again and again and again and again and again. You can wait for all those channels to open and close, and finally make it to the very end of the axon terminal. You can speed this up by something called myelin. This increases the speed of the signal transduction along the axons by saltatory conduction of action potentials. And so this is the myelin, this yellow stuff right here. And this thing between the myelin is called the node of Ranvier. So you're gonna shoot. This is like the myelin is like insulatory stuff. So you're not gonna lose any charge. So let's say you get an action potential, shoot it up. Okay, this positive charge is gonna shoot down all the way because you can't have any change of potentials here. So it's gonna go all the way down until the node of Ranvier, where there is no more insulation, and then that's where you can get a new action potential. There's a ton of sodium channels in the nodes of Ranvier, and you're going to get another action potential that's going to shoot all the way down here, get another action potential, again and again. The way I think about this is basically I think about it as like a car. Car with gas. Gas is the voltage, okay? You have a little bit of a leaky car, it's leaking gas, so your voltage goes down as you go. If you leak too much, you lose all your gas, you gotta fill up, slows you down. If you get myelin, it like kind of plugs up your little leaky gas tank. You're not leaking gas, you don't have to stop as much, you don't have to make as many pit stops, you can get to your destination faster. That's what I think about this like. It's the myelin is basically a stopgap to prevent your gas from leaking, lets you go further. So this increases your conduction velocity, your signals go by faster. You also get increased space constant. Basically, your one tank of gas is going to take you a lot further. So your increased space, your space constant goes up. It's the amount of distance your signal can travel, because you're basically, your myelin is the insulator, it prevents loss of the voltage, and decreases time constant. Basically, your signal travel. What it is actually time constant is it's a little complicated. You don't have to memorize this if you don't want to. A time constant refers to how fast your voltage across the cell membrane can change. Remember how I said the nodes of Ranvier have tons of sodium channels. So they have very high. So this is a node of Ranvier. very high sodium channel, so you can get the depolarization very quickly. You can get a ton of sodium ions into the cell very quickly. So it's very short time, so it's a short time constant. Finally, remember that in the CNS, we already talked about this. What makes the myelin in the CNS is the oligodendrocytes, and what makes myelin in the peripheral nervous system, that's the Schwann cells. Alright, so next we're going to talk about what goes wrong in with myelin.